This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 213, recorded on December 28th, 2012. Hi, everyone. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How are you at the end of this year, Alan?
Doing okay.
Yeah.
Window of nice weather today, or nice-ish weather. It's clear skies anyway. We got some snow and some slush, and then it's supposed to get really, really cold.
Pretty nice.
But today
here.
It's nice.
Pretty nice here. Blue skies. It's kind of cold. 3 degrees Celsius. Let's find out what the weather's like in the rest of the country.
Yes. Let's go counterclockwise, right?
Okay. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
I got the counterclockwise right.
Good job.
Yes.
How's the weather out there?
We have 27 degrees Fahrenheit, -2 Celsius, and it's kind of sunny right now, and we had sun yesterday after our 7 or 8 inches of snow. So that was nice, because in Michigan, we have a lot of cloudy days because of where we are between the lakes in the wintertime. So,
Did you make a snowman?
No, it wasn't that, wasn't the right consistency for snow.
Oh, my. If we waited for consistency, we'd never have a...
Well, you can waste a lot of time trying to make a snowman if the snow isn't right.
I suppose.
Yeah, if it's really powdery or if it's too wet.
Yeah.
Of course, where our next host is, there is no snow. Rich Condit, how you doing from North Central Florida?
Great. Hi, everybody.
Hey, Rick.
65 degrees Fahrenheit, clear blue skies. That's 18 Celsius. So it's a nice day.
This is the season when Florida justifies its existence, right?
That's exactly. Exactly
I
right.
don't mind the cold weather. You can build a fire. That's what we've been doing, you know.
Yeah.
Got a lot of wood around.
Oh, yeah. Yeah.
A couple telephone poles, power poles.
Oh, yeah. Most of the poles they took away, but there's plenty of trees left over from that stuff. Yeah.
I still have about 3 cords split and stacked in the back from last year's October storm.
Yeah. We're still trying to figure out now. They're raising the flood levels at the beach, you know, for insurance purposes and all this stuff. So they're giving new numbers about if you're X feet off the ground, you have X% chance of being flooded each year. So all the elevations are going up. So if we put our house 8 feet above the ground, we'd have a 1% chance of being flooded every year. So, our house is maybe 5 feet off the ground. The thing is, if you lost half your house and you want to rebuild it, you have to rebuild it to the new specifications. So you'd have to put it up on piles. 8 feet is pretty high up.
See, I think they ought to just go through and buy out all these properties that got flooded at their pre-flood price and leave it as empty beach.
Hey, I'd sell.
And if you did that after every major disaster, you would not have to bail the place out the next time, because that's what's going to happen. Everybody's going to rebuild. They'll build to the new spec, and then another flood will come and do a lot of damage, and then they'll have to rebuild to a new spec.
Yeah. I'd buy out. I'd sell out at the pre-flood price, though, not now.
Oh yeah, absolutely. It's
because the work that we have ahead of us.
I think it would end up being cheaper in the long run, but I'm not in charge of these things.
Oh, anyway.
Why not? Why aren't you?
Why not? I always ask myself that.
Why aren't you in charge? Okay. I noticed people are surprised that you're so tall, Alan, or so short.
So short, yes. Apparently my bark is worse than my height.
I wanted to set you up.
Yes.
Okay, it is the end of the year. And by the way, since it is the end of the year, we have our by now traditional year-end review, which here at TWiV is very special, of course, because we're reviewing something which is just so cool, and that's viruses. Before we do that, we have a follow-up from Robin who gave us a correction CRNA is Certified Registered Nurse Anesthetist. They sometimes make as much or more than some primary care MDs.
Which is no trick, by the way.
No. CRNP is Certified Registered Nurse Practitioner, which he talked about last time. Okay, because he had mixed up the terms. CRNA and CRNP, which are terms from virology, by the way.
Yes.
So, let's see. We're going to pick 12 stories this year.
So they need a certified designated nurse anesthetist.
Yeah, CDNA.
Right.
You're right. Yeah, we could go on with that one.
Yes, that could go for a while.
All right, so I thought we would pick 12 stories because there are 4 of us. You know, if we did 2, there'd be 8. That's kind of a short list. And, well, so there we go. That's why 12.
And it's 2012.
And it's 2012 and it fits. But next year we're going to still do 12 because I'm not going to do more.
Who knows what will happen next year?
We don't know.
There may be, 4.2 of us.
Right.
You know, one thing you can't do is predict the future, Rich.
That's true.
Well, I guess now that I've disclosed my height, I could be one of the fractional members.
Let me give you a summary. We did 52 episodes of TWiV in 2012, including one that I'm going to release tomorrow. It's a present for everyone, which no one knows about.
Oh.
And
nice.
Even we don't know about it.
Even you don't know about it. So maybe you'll be surprised. And that started at 164 through 213, which is this episode. So that's cool. 1 a week.
Yep.
We traveled to Dublin, Glasgow, Chicago, Madison, Stores, Boston, and Lincoln. 7 trips. One of those was with 2 of those, right? 2 with Rich and Boston, of course, Rich and Alan. And Rich and Alan got to meet in Boston for the first time.
Yes, in person.
Yep.
Where we learned that Rich is quite tall.
That's right.
You really have a problem with this. Is that right?
No.
Alan? Would you
I'm
like
just
to
filling
talk to
people
somebody?
in. It came up on Twitter. There were comments about Alan. I thought you were 6'4" or something based on your voice. I'm 5'9".
I'm the shortest.
So I don't know what
worry
height people
about it.
expected Rich to be.
He's the tallest, right?
Yeah.
We had guests on 28 episodes. Over half of our episodes had a guest. I don't count you as a guest, Kathy, even though early in the year you were.
Okay.
I figured you're a host, so.
Okay.
56 guests total. It's not bad for a hobby, is it?
Great.
Not bad at all.
Terrific.
I mean, none of us is this our primary job.
Right.
So I think it's pretty cool. We really are into it, so.
And other people are too, that's the neat part.
Yeah, that's very good. We have lots of listeners, and that's great. And hopefully 2013 will be just as cool. I'm sure we'll have plenty of cool stories because viruses never fail. There's a title.
Viruses never fail.
Actually, they do sometimes. They do a lot, probably more than they don't. Yes.
They're not going away anytime
Not going
soon.
away. All right, in no particular order. Is it okay if we just go down this list as written here, everyone?
Sure.
And you can chime in, of course, as always. Well, I have at number 1, H5N1, which we covered in 5 different TWiVs, 168, 173, 177, 182, and 190. Avian influenza H5N1. This was a brewing story at the end of 2011. Just to review, 2 groups had done experiments selecting for variants of H5N1 that could travel through the air and infect ferrets. And these manuscripts were reviewed by the NSABB. What's that stand for again?
National Science Advisory Board for Biodefense.
Biosecurity.
Biosecurity, thank you.
Yes. And they decided that they shouldn't be published.
And to make another point about NSABB, it was not their original mission to do this sort of thing.
Not at all.
What were they supposed to do?
I don't think they were necessarily supposed to review manuscripts or.
Well, when Michael Imperiale was on the show. That's one of those episodes you just listed.
Right.
He went over this, and they were convened to, as one of these advisory committees, they were supposed to get a group of experts together and solicit outside input as well, and come up with guidelines for how to deal with this dual-use research of concern, as they call it, studies that have a peaceful purpose, but that could possibly be misused in the biological sciences. And so they were supposed to be, and they were, a committee that met and discussed these issues at a high level and produced reports and recommendations and that sort of thing. And then one day they suddenly had these manuscripts dropped in their laps and were told, tell us what to do with them.
And that's an important point. They didn't go hunting for this stuff. They were asked to review these.
Yes, this
was
was
a matter of
sent
fact,
to
if
them.
I understand it correctly, they were asked by, among other things, the editors at least one of the journals that was involved,
Yes.
who didn't know how to, saw a potential dual use issue and didn't know how to deal with it, and turfed it to the NSABB.
Right. Well, I think turfed it to NIAID, which turfed it to the NSABB.
Yes, right. Okay.
There
Fair
wasn't any mechanism for dealing with this, so they grabbed whatever was handy, and that was this particular committee.
Right.
Now, eventually, the papers were rewritten, and NSABB looked at them again, and then they voted to publish them.
And I think, and importantly, in the meantime, there was a meeting of a number of people at the World Health Organization.
Yes.
Who their own read on the thing was that the papers did not need to be redacted, that they could be published.
Right.
And that may have had an impact on this whole thing.
I think what also had an impact was that there was a much broader discussion among virologists who hadn't been in on these initial deliberations about the risks of redacting research, which was something that hadn't really been fully explored in the conference calls when they made these decisions. And I think probably another factor was that the re-review after the papers were revised took place in an actual in-person meeting rather than conference calls.
Well, we were part of that discussion, clearly, since we covered
Yes.
it so much. Now, when the papers finally got published, we talked about them on two separate occasions, one for the Fouché, one for the Kawaoka manuscript. The results are really interesting, and as I said, and we couldn't imagine not publishing them. This way, scientists can discuss them.
Or deleting key parts of them, because it was
Terrible.
really,
Terrible.
I mean, the essence of this was that the virus can mutate and adapt in ways that are informative.
Now, what really became clear was that Fouché had really exaggerated the findings in his talks with a journalist way back, I guess in August or September of 2011, because he had said they can transmit through the air among ferrets and they are lethal.
Right.
And, of course, in both cases, it turned out that these viruses were not lethal after airborne transmission. They had become attenuated. And so, this continues in my mind to be a problem, why that was so, and why it was so misinterpreted or misrepresented. And Martin Enserink, who's written a lot about this, so I had written a blog post and accused Enserink of somehow misrepresenting the situation, and he actually posted a comment saying, "No, I didn't misrepresent anything. Fouché, that's what Fouché said." So I'm glad he did that.
Well, in the digital age, you got to be really careful what you say. You have to be as accurate as you can. You have to understand that a lot of people have access to whatever you're going to say.
Sure. Now, the story is not over. Of course, there's still a moratorium in place, which was established at the beginning of this year, which is a moratorium on transmission research on H5N1 strains. And the U.S. government is now putting together a draft of some plans that would regulate research of this sort. There's a very nice Science Insider article that we will link to on this. But among other things, there's a list of there's 7 criteria that any kind of H5N1 study would have to meet to get NIH funding, say. And some of them are very difficult, and one in particular. Number 2, the research does not intend nor is reasonably anticipated to yield a highly pathogenic avian influenza H5N1 experimental virus which has increased transmissibility, pathogenicity, or expanded host range unless there is evidence that such a virus could be produced through a natural evolutionary process in the foreseeable future. This is such a ridiculous statement. I don't know how any scientist would be happy. And according to the comments in this article, many of them are unhappy with this.
Yeah. I don't think that's going to survive, or at least it's not going to survive in that form. There's too many people who say, "Oh, wait a minute, your buddy down there in Gainesville,
Ken Burns, says
right.
The idea that we can do something that nature can't doesn't stand the test of credibility."
Right.
I like that very much.
Right. And the idea that we can predict what's going to happen naturally, especially with a flu virus.
Right.
They have a new chair of the NSABB, by the way. So Paul Keim is gone. The new chair is Samuel Stanley, who's president of Stony Brook out in Long Island. And he says, my read of this is that it would really put a stop to most of this research. I'm not sure how one would get that evidence. I think it sets a bar that may be too high, in my opinion, to allow you to do any gain-of-function experiments. While I appreciate the risks, they are very powerful tools when used appropriately. I think this sounds like a really good chair of this committee.
Yes, that was an encouraging quote, yes. Well, what this has done, it seems to me, is to open up again the discussion that started back in 2003 when the NSABB was created, which is to create a genuine mechanism for reviewing stuff that has potential dual use. Okay?
Yeah. Right. And finally, in the same article, Peter Palazzi, my old friend here, says So the problem is that the NSABB is targeting gain of, not NSABB, but NIAID or HHS, is targeting gain-of-function experiments on H5N1. So Peter says gain-of-function experiments are almost always loss of function for another property. In this case, you gain airborne transmissibility in ferrets, you lose pathogenicity. Thus, the NSABB looks only at one side of the coin. Anyway, so there was a public discussion period for this, and at some point they're going to decide what guidelines to adopt, and at that point probably the moratorium will end, because basically if you're going to do any of these experiments, you won't be able to get HHS money, which I think is really unfortunate. I think we can learn a lot from this, and I think they are low probability that something bad is going to happen, but the benefit is high, so I really don't understand the fear and the necessity to restrict them. And of course, this is for the U.S. Who knows what the rest of the world is going to do? So,
right, which could, depending on who does what, could largely render this moot.
Right. I mean, you can imagine Ron Fouché is going to do as he wishes because he won't be bound by this unless he uses NIH money, of course.
Yeah, you'll get talent fleeing to other agencies, other granting agencies that will provide the funding for it, and those could be international.
My guess is that in the long run, much the same as with the recombinant DNA debate, there will be little, if anything, that's actually restricted. There'll be, a sharper eye on what's being done and a little more care in presenting it, but the actual restrictions, I'll bet, will be few to none.
I hope so, because it makes no sense to me to restrict what you can do. If you want to contain it, that's fine, right?
Oh, yeah.
But to not give you the money to do the transmission experiments, that assumes that you can predict everything that's going to happen, and that's obviously contrary to what we do in science.
Yeah, I have a big problem with tying the grant money to some kind of predicted outcome of whether it'll be dual use, and I have a huge problem with this idea that there could be a retroactive classification of research that started out unclassified. That's just dumb.
What do you think, Kathy?
Well, I was just going to say that I've brought this topic up to several friends of mine who are scientists or well-read, but not necessarily virologists. And this whole story had escaped some of them, which I know we all find hard to believe. But somehow, a big portion of the story didn't get out to a lot of people. That just was really surprising to me. And I mentioned it to someone who follows Twitter, and that portion of the Twitterverse that he follows, he said there wasn't really anything there about it. So it just was a different perspective. It was kind of eye-opening to me. It's happened in probably 3 or 4 different conversations.
I think the general public perception of this, if it existed at all, has vanished completely.
Right.
Which is
This
interesting.
completely off the radar.
There were a couple of huge front page splashes in several newspapers.
Right.
Right, but people, when you have a huge front page story on a science issue, or really any issue, this happens in politics, in just about any topic. The public memory for it is a few months. Yeah. And there's some vague notion that something happened with flu at some point, but nobody can tell you exactly what.
Right.
Okay. My next pick was polymorphisms that control susceptibility to virus infections. We did 2 TWiVs on this. TWiV 175, we talked about mutations in the innate immune system that predispose children to herpes simplex encephalitis. And in TWiV 180, we talked about mutations in a interferon-stimulated gene that also are associated with severe influenza. And so I think this is just the beginning. I think we're going to see a lot more of this as it's becoming cheaper and cheaper to sequence genomes. People are going to be looking for the reasons why certain people not only get serious virus disease after a virus infection and others do not, but the whole spectrum of response to an infection, mild, intermediate, serious disease. I think people will try and correlate with specific mutations. And the two cases we talked about are pretty straightforward. There's a mutation in a gene that knocks out the gene, basically, and makes you get worse disease. But there are going to be more subtle associations, single amino acid changes that affect the interaction of various proteins, harder to tease out. But I think this is a big direction that our field is headed in, to try and understand, why someone down the street never gets sick and someone else is always getting sick. So I would say that in the next couple of years, this is going to be a big one. I'm particularly interested in this. I think this is really cool. For example, I think people who get vaccine-associated polio, I think they have some mutation that makes them susceptible, because most kids excrete virulent vaccine strains and they're fine. And there's 1.5 million that get vaccine-associated polio. What's the difference? I don't know if we're going to be able to figure that out as the disease is eradicated, but I think that's just one of many, many examples.
Yeah, I was going to ask if there was enough material around stored or something someplace so that you could address that issue.
There probably is. It's just a matter of getting someone interested in doing it, because if the disease is going away... Yeah, they're going to say game over. Yeah. Spend your money elsewhere.
But it's not just about polio.
Right.
Of course.
I mean, that's something that if you can find what controls susceptibility to that, it comes back to polio being a great model system, because you've got the decades and decades of research on it. It's extremely well characterized for a human virus. And if you could get at what's driving the pathogenicity in those cases, if it's some specific allele, or if it's something in the gut microbiome, which would be a little harder to get to, but if you could dissect that out, I think it might give you some insight on other diseases too.
Sure.
Right. It goes along with what I've been saying for years, and that is that relative to what we know about microbial genes involved in pathogenesis, so virulence genes in bacteria, viruses, or whatever, we know very little about susceptibility genes in the host. And I think a large part of that is due to the genome sizes of the microbes relative to the hosts. But even in the small little corner of what we've been doing in my lab, where we identified a gene for susceptibility to mouse adenovirus, or a gene region that encodes some of the Ly6 genes, those Ly6 genes have been implicated in susceptibility to HIV, West Nile virus, herpes virus of chickens. And so it goes along with what Alan was just saying, is that you might find out things that are widely applicable, not just to one or two viruses.
Do those genes tell lies?
Ha! Ly6, not Li.
It sounds the same.
Yes,
Well,
it
if
does.
it's in mouse, then it does lie.
Right.
That's right.
The monkey version just exaggerates. But this also ties into something that's become a big deal in the pharma industry, which is this idea of idiosyncratic responses to drugs.
Mm-hmm. Right.
And it's a little bit of a tangent, but it's kind of the same issue, that you give the same thing to 1,000 different people and 999 of them respond fine, and the 1,000th one is dead, or something like that, or you have some drug that would be a great drug if you could fit, anti-clotting agents come to mind. Warfarin, figuring out somebody's proper warfarin dose is this huge deal, because you have these idiosyncratic responses that can go on that can be lethal.
Right.
And I think there may be similar mechanisms at work in those things. So it's a hugely interesting field, and you're right, Vincent. It's finally to the point where we've got the tools to look at these.
Now, it's not easy, because if you do the whole genome, you get all this noise. And
Right.
You have to sort that out from what's really doing something. So unless there is a loss of a protein, it's harder to do. So
Right.
the herpes encephalitis was a truncation of a protein, so that's easy. But it's hard. It's a lot of data. It's millions and millions of bases, and
Yeah.
this is where the bioinformaticians come in.
Yes.
Okay. And my next one, I, of course, started my lab here working on a virus receptor, the poliovirus receptor. And so those, even though we don't do that anymore, those have a special place in my toolbox. So we had a couple of stories on new virus receptors this year, some big ones. Of course, the most recent one was the hepatitis B virus receptor that we talked about in episode 210. A beautiful story out of China, using a little peptide to pull out a protein from cells and then identify it from tree shrews.
Right.
And this turns out to be a sodium taurocholate transporter. Also, we talked in 166 about new receptors for hepatitis C virus and Ebola virus and measles viruses. 3 in 1. That's why we called that episode Breaking and Entering. The hep C is Niemann-Pick C1-like protein, and Ebola was Niemann-Pick C1. The Niemann-Pick C1-like, which we had gotten wrong, we called it C1, but they're different proteins, is a cholesterol absorption receptor, and there's actually a drug, an antagonist of that protein, which is FDA approved, and that was shown in the paper, among other things, to block hep C uptake. So it's a cool experiment.
And these are some of these are not receptors in the classic sense of bind the virus at the surface being their primary function, but rather a lot of these are post-entry molecules, right?
That's right.
Right.
So the virus can't complete its entry because it's got to travel in endocytic vesicles and et cetera, and do a bunch of stuff in the cells, and requires cellular components for that. So there's going to be a slug of these down the road.
Yeah, the C1 receptor for Ebola is something that works in the endosome. It allows fusion between the virus and the endosome. So you're right, it's not a surface receptor. We don't actually know what that is for Ebola. I recently spoke with Kartik Chandran, who is an author on this Ebola Niemann-Pick C1 paper, And they're trying to get the cell surface receptor for Ebola. So maybe that'll be another one for 2013. We'll keep an ear for that. Anyway, I think these are really, we know a lot of the virus receptors. There were a few unidentified, and these are some of them. So I thought those were good stories, and I'm sure we'll hear more.
I'll be interested to see how many other of these intracellular guys show up. There'll be a bunch of them, and I'm also very interested to see what develops out of the hepatitis B receptor. That's going to go a long way. That opens up a lot of doors.
Yeah.
Mm-hmm.
All right. Our next one is called One Last Nail in the Coffin for XMRV. And that made me think of John Coffin.
Yes.
Whose lab was instrumental in showing, of course, that XMRV, originally identified in prostate tumors in 2006 and then associated with chronic fatigue syndrome in 2009, was in fact a laboratory contaminant produced when a human prostate tumor was passed in mice. The virus came from the germline of those mice and recombined and multiplied in the human tumor. And we had a TWiV special with Ian Lipkin. In my view, this story was pretty much over at the end of 2011, but we were waiting for this multi-center blinded study, which Ian was coordinating, where they had a number of labs pick specimens and look at them in a variety of ways, PCR, cell culture, antibody assays, and look for the presence of XMRV or other related retroviruses. And that study was released in September, and as everyone probably knows, it found no evidence for the presence of this virus in any of the samples from either CFS patients or controls.
The title
As
is
expected.
The title is kind of a short abstract, basically.
Yes.
A multi-center blinded analysis indicates no association between chronic fatigue syndrome, myalgic encephalomyelitis, and either xenotropic murine leukemia virus-related virus or polytropic murine leukemia virus. That's the end of the title.
Just in case you were wondering what the paper was about.
And I love the phrase that they used in the paper, that this they wanted this to be a paradigm of pathogen de-discovery.
Mm-hmm.
Yep. In fact, that's what Ian had said, and that was the title of that TWiV special. Now, if you remember this...
I still think the study was probably a waste of money. Well, no offense to Ian. I think he did an outstanding job in the situation, and Tony Fauci asked him to do it, and, of course, you do it. But I think that this was not necessary for that conclusion.
Well, it's, it got all the groups together, and they all agreed in the end that there was no involvement, so that may be worth $10 million. I don't know.
So that's the issue I have with it.
I understand.
The quantity of money in the funding environment that we had.
I understand. I fully understand. Also, we had wondered, in fact, I asked Ian in that special, what about Prostate Cancer Association? There were a number of papers out there saying you can find this or not in prostate tumors. And not too long after that Lipkin study was published, there's a wonderful paper published in PLOS One. And it was called In-Depth Investigation of Archival and Prospectively Collected Samples Reveals No Evidence for XMRV Infection in Prostate Cancer. And the authors on this paper include all of the authors on the original 2006 paper from Joe DeRisi's lab, where they had identified XMRV from a prostate tumor. This is an amazing paper. We never covered this on TWiV, but you should go have a look at it.
Well, we did cover Ela Singh's paper, which was also a really amazingly thorough analysis of what turned out to be a contaminant.
So that was CFS, her paper, though.
Yeah, her paper was CFS, but of course it called into question because she found the contaminant in her lab that was the source of this. That called into question all of her prostate cancer data as well.
Right, but no one up until this point had retracted any of the prostate data. So this paper basically spurred that on. They got prostate cancer tissues and blood from 39 patients, and they got archival RNA and prostate tissue from the original DeRisi et al. 2006 study. They basically found no evidence for XMRV anywhere except in the archival RNA, which they found had been contaminated with XMRV from a cell line because they found mitochondrial DNA from that specific cell line contaminating the archival RNA. Really a wonderful story. Now, the interesting thing here is that when this paper was published in PLOS One, the journal editors retracted the original paper without having the authors agree to that.
And that was from PLOS Pathogens, right?
Yes.
So
because the original was in PLOS Pathogens, right? Yeah.
Right.
And I'll put a link to an article in Retraction Watch about this, which kind of summarizes everything. I mean, basically, the editor-in-chief said, well, we tried to contact the authors and we didn't hear from them, so we decided after a day to retract it. I just think that was crappy because they could have waited to get the authors, to do that. Because it's always better when the authors retract it, right? Mm-hmm. So I didn't like that at all.
Yeah, and I don't know. There's this whole debate that this and some other recent events have raised. What justifies a retraction?
Yeah.
Do you go back to all the literature and say, okay, every time the paper got the wrong answer, we retract it?
No, I don't think that's right, because half of all science would be retracted, wouldn't it?
Right, but then you have to say, well, how wrong does it have to be? And I'm not sure it was appropriate to retract this paper because anybody who looks this paper up is going to see that it was referenced from a later paper, and of course you would look at that later paper and find out, hey,
that's right.
This is what the story turned out to be.
I mean, I think that fraud should be retracted.
Yeah, I think fraud or really, really grievous error in the data itself.
Right.
Where there's something that, hey, we totally screwed up how we did this experiment and we ended up publishing it. But fraud, I think, would be the primary reason to retract.
Now, in the meantime, of course, chronic fatigue syndrome still exists as a disease, and we still would like to take care of it. And a couple of follow-ups there. Just recently, a drug called Ampligen, which has been used to treat patients. Ampligen is basically double-stranded RNA, and it was given a clinical trial by a company called Hemispherics, which is a Philadelphia-based company. And a FDA advisory board very recently met on December 20th, and they decided basically that it didn't really look good. So they voted on 4 questions. One, is there substantial evidence of efficacy? So this was used in a clinical trial with CFS patients, of course. The vote was 9 no and 4 yes. Has safety been adequately addressed? 9 no, 4 yes. Is the safety profile adequate for approval? 8 to 5 yes. Do the safety and efficacy data support approval? 9 to 4 no. And an industry person, Brian Kotzen of Amgen, said that the inadequacies of trial design and irregularities in the data didn't measure up to the industry standard for showing safety and efficacy. So this is not binding for the FDA. Their final decision will come up sometime next year, but it doesn't look good for this drug unless they do some more work. Meanwhile, there was a paper earlier this year in PLOS One, which showed in a Swedish population benefit in CFS patients using a drug called rituximab, which is an antibody against the B-cell protein CD20. And that's generating a lot of interest. So we'll see where that heads.
Right.
That
so
strikes me as... Go ahead, Kathy.
I was just going to say what any of these drugs that end in IMAB tend to be monoclonal antibodies, just for those of you who may not have picked up on that. And, using this antibody depletes the B lymphocytes, the B cells in these people. So the interesting thing is that there's pretty rapid B cell depletion, but there's a delayed response for the CFS symptoms or signs, I guess, which suggests that CFS is an autoimmune disease. And so basically there's some gradual elimination of the autoantibodies that precedes the clinical responses. So,
which makes it puzzling to me why giving them double-stranded RNA would help, because it's stimulating an immune response. But
right.
You know, they don't actually know how Ampligen works, so who knows what's going on.
Right.
Anyway, there's obviously still a lot of interest in CFS, and I think it's going to be a tough nut to crack.
Yeah.
Okay.
Yeah, measuring the outcomes in these studies is very difficult, and defining the patient populations is very difficult, and picking the drugs is very difficult.
Right. All right, Kathy, you had the next one.
Well, I picked the fact that vaccination programs, particularly for polio, have been in the news a lot lately. So just to remind everybody that there have been longstanding efforts to eradicate polio. There are 3 countries, I think, where it remains endemic currently: Pakistan, Afghanistan, and Nigeria. And most recently, there was this story from Pakistan of vaccinators, as they're called, being killed for their activities in promoting the polio vaccinations. And immediately I thought of the story that Alan blogged about that came out after bin Laden was killed, where the CIA had put it up or had started a fake vaccination program for hepatitis B in order to try and collect DNA samples. And they did this in the geographic area around bin Laden, so that they could perhaps get some DNA evidence that, yes, he was residing in the compound. And I think the physician that was involved in that was later killed, right? Or at least
he was convicted.
I think he got a 30-year sentence, something like
that.
Yeah,
he was convicted and imprisoned.
Yeah.
Okay, so in any case, setting up this fake vaccination scheme, I think, could have led to some of the distrust that there is about vaccinations for legitimate bona fide polio vaccination going on. Or even if there wasn't a direct connection, there's enough misgiving and rumors about vaccines that doing that to try and get the DNA around the bin Laden family was maybe detrimental. Anyway, so
I would certainly agree with you that it was not helpful.
Yeah.
It was it was not what I would have preferred the CIA to do for the sake of public health, but in that case they were not focused on public health, obviously.
Right.
They had a particular target they were pursuing, and they had good reasons for doing that. So I'm as I said on my blog, I'm deeply conflicted about this. I really don't like that it was done, but I can't really say that it was necessarily as evil as some folks have made it out to be.
Which the CIA fake thing.
Yes,
right.
the fake CIA, which apparently from the information that's been made available, as far as I can tell, they actually distributed real HBV vaccine.
Mm-hmm.
So they did actually vaccinate people, but they had an ulterior motive, which was to collect DNA from members of the bin Laden household.
Right.
And in this recent round, in this case where the vaccinators were killed, my objection to the coverage is that it's focused heavily on that particular incident, which misses the broader context of what's going on. Pakistan's a very complicated place. And there you have a government that's very unstable. You have a military that's on the brink of a coup, I think. And there are these fundamentalists who are kind of half running the place. and I don't think it's a coincidence that most, if not all, the vaccinators who were killed were women. That's very threatening for the fundamentalists there, who have this very reactionary view of the appropriate role of women. And they, I don't think that's a coincidence that these women in public health who have an authoritative role in it were the targets for assassination. And the Taliban, which is claiming credit for this, I believe, has been complaining mostly about drone strikes.
Right. I seem to recall something some time ago about the Taliban saying that they were going to target the polio vaccination program unless the drone strikes were stopped.
Yes.
So it's basically an easy, high-profile terrorist target.
Yes.
Very high profile.
And very, very poorly protected.
Right. The most recent article in the New York Times indicated that in some modern medical surveys, not necessarily polio campaigns, teams do carry GPS devices so that they can find houses again. And then the Taliban makes a connection. Well, drones use GPS coordinates. So there's just a lot of potential here for rumors and all kinds of crazy sounding rumors that have come out about this in the past. That the vaccine contains pork, or that the virus itself causes AIDS, or is a plot to sterilize Muslim girls. All of those things have to be overcome, and adding in, potentially this vaccination to get DNA scheme, even though, as you said, Alan, it was a valid vaccination campaign as well, I think just doesn't bode well for polio eradication.
As I say, it didn't help.
Right.
But the bigger problem is actually Nigeria, where at least some of this paranoia probably originated. Some of these same sorts of rumors of the vaccines plot to sterilize Muslims and this sort of thing. That was and I think still is a very current belief among many people in Nigeria, and that's been the source of I think it still is, right, Vincent, the majority of new polio cases?
Some of them. Now there's so much still in Afghanistan and Pakistan that can be sources of that as well. In fact, complicating this whole story, there has recently been an outbreak of vaccine-associated or vaccine-derived type 2 polio in Pakistan.
Right.
And I talked to someone about CDC, and he said it probably originated from Afghanistan and has been circulating for 2 years. So it's not all from Nigeria. A lot of it is, but not all of it.
And the vaccine-associated cases are certainly not helping the PR effort either.
No, that could be
a problem. I mean,
then you can actually make a true statement that the vaccine is, it can cause disease.
Right.
Yeah, I mean, this is a big problem because this is the vaccine of choice for the eradication, yet we know it can cause polio. But, previously there's been so much wild polio that those cases were not an issue. But now as the numbers get less than 200 per year, these vaccine, derived cases make a difference.
Yeah.
And so they had, I think, 10 in Pakistan, and then it spread over the border into Afghanistan. There were 2 cases there. And the solution is, well, part of the reason this arose is that they haven't been immunizing against type 2 polio for a while. They've been using types 1 and 3 because polio type 2 has been eradicated, quote unquote, from the globe since 1999. However, the vaccine strains circulate for long periods of time, and if you stop immunizing, they come in and cause polio, and that's what happened here. The solution is to go in and immunize with type 2 vaccine. But here the problem is that because of the Taliban's influence, thousands and thousands of people are refusing to be immunized because the Taliban tells them not to be. It's a real problem.
So, Vincent, you have a nice blog post about this, too, that you should include in the show notes, I think.
Yeah, that's where Alan had made a comment, actually.
Yeah.
All right.
Yeah, and I'm not, just to be completely clear, I'm not saying that the CIA vaccination ruse had nothing to do with this.
Right.
I think that probably is a factor, but I don't think that it's the only factor.
Yeah.
Right. Well, it I think back to when I remember hearing that polio was going to be eradicated in 2000, and that is now a long time ago. So,
and this illustrates why, in part, it's hard. It's not just getting the vaccine to work and immunizing, but there's other things as well that factor in.
Getting people to behave rationally.
Yeah. Yeah, Kathy, you had the next story as well.
So, yeah, I really thought that there were several discussions about the funding situation in science. Jonathan Udell had his, which was in TWiV 208. There were also the articles by Arturo Casadeval and Ferric Fang that came out, I guess, officially in early 2012. Their dateline is 2011. Those were linked to in TWiV 184, and you guys spent some time talking about that. And what I thought was interesting is that John Udell has some pretty interesting ideas about how we might try and reform science. And although it might be hard, there's the people that are willing to make some efforts at trying it. And his recommendation was to perhaps try something like the NIH intramural model, where labs are not of a huge size, and there are people employed that are staff scientists that get paid a decent wage, something above what postdocs get paid. He had an idea that indeed probably we have trained too many scientists, and he thinks maybe we should train half the number but train them twice as well. And then, but also that not everybody that gets trained as a PhD necessarily should be a PI, and that's obvious to anybody who's been around academic science for a while, realizes that we have lots and lots of trained postdocs who aren't getting academic positions. But what it enables you to do is think logically and learn how to present yourself. At the same time, you're learning biology, you're learning how to evaluate evidence, and so you can have some kind of career in science that, again, isn't necessarily being a PI, but is somehow making use of this education that you've gotten. And so here in our combined graduate program, there's an effort afoot to try and address some of these issues. To some extent, we're taking fewer students into the graduate student pool in this coming year. That's not entirely because of this kind of reasoning, but also this group has gotten together to talk about some things presented by Keith Yamamoto and others about not just calling these alternative careers, but just careers in science, talking about a diversity of careers, a variety of careers, so that there's not a stigma to having a career in science that isn't being a PI.
Right.
So I think that kind of discussion and dialogue is important. So that's why I thought John Udell's TWiV was really worth listening to.
Yeah,
I like that. I like that bit about removing the stigma by removing the emphasis on alternative careers, because I thought of myself as pretty unbiased, but I'm guilty of using that terminology as well. That's very revealing. That's very interesting.
Yeah,
and that's somebody pointed out that the majority of the students now going into graduate school are not going to be PIs.
Mm-hmm.
So we're not talking about alternative careers. We're talking about the main careers that they're going into are other things. Being a PI is an alternative career.
Yeah.
right.
One thing I wanted to add to this, because it's sort of in the same vein, is a paper that I discussed yesterday on TWiM with the author Joe Handelsman, who's a professor of microbiology at Yale. And this paper was published in PNAS earlier this year. It's called Science Faculty's Subtle Gender Biases Favor Male Students. Everybody needs to read this.
Yes.
This is an amazing study. They sent out a letter which summarized the qualifications of an undergraduate for a lab position. And they sent it to 127 science faculty members, professors, half male, half female. And they were randomized. Half of them got a letter where the person was a John and the other half got Jennifer. Otherwise, exactly the same CV. And they were asked a series of questions. whether they'd hire this person, how much they'd pay them, whether they would mentor them. And very clearly, the guy won most of the time. The guy was going to get hired. He was going to get more pay. He was going to be mentored more than Jennifer. And this is equally across male and female professors.
That's interesting.
So this is called subtle gender bias. It's subtle because it's not overt. You're not doing it because you don't like women, for example, and they built in some questions to get at that as well. And Jo is very into this because she's part of this Presidential Council of Advisors on Science and Technology, and they say we need a lot of scientists in the next decade, not necessarily professors, but all kinds of scientists and engineers, and we don't have enough, and women are a great pool, but we have this bias that's going to prevent them from going into science. This is really stunning, this finding, and both Jo and all the people involved were stunned to actually see. The results are so clear, you don't even have to be a statistician to see them. So I think that goes in hand with your career sort of picks, Kathy, because this has got to go. We have to figure out how to fix this.
Yeah. And
So-
go ahead.
Well, I was just going to say I've been involved in a program here at Michigan, the Advanced program, that was started originally by an NSF-funded program and has been continued on by the university. And in this group of people, we got together and read all kinds of papers. And I'm not going to downplay the findings of Jo Handelsman's paper, but there are a number of papers along this line that have been out there in the psychology and economics and sociology literature about, they're mostly what I call CV or resume studies. And you send out resumes and you change the names. One has an African-American sounding name and one doesn't, but it's the identical CV. And there's a bias toward the non-African-American sounding name, or men and women, just like this one, or even LGTB kinds of CVs and resumes where they're identical, but there's some reference in there that makes you think that they might be involved in a gay alliance or something like that. And so this is just one more piece of evidence that we have these unconscious biases. And again, the big surprising thing, I think, to many of us is that this is true of men and women who are doing the evaluating. We all have these biases, although we, profess not to have them consciously. They do creep in, and so as Virginia Valian says, she's written a whole book on this. These are little molehills that just pile up and become mountains over time. So,
yeah.
Well, part of the problem here is that in terms of mentoring, the women were at a disadvantage, or Jennifer was at a disadvantage. Most of the time, the professors didn't want to mentor her, had they even accepted her, and that discourages them from going into the field at all. So it has a long-range effect.
And what I found really interesting about this study was not so much the study itself, as Kathy just pointed out. This is part of an entire long, well-established genre, but the reception of it. And come on, we've known for years that women are at a disadvantage in science. You look at the faculties of the top universities, and there's something going on. And it's not that the women are not good at science. There's clearly a problem here, but this puts a very specific marker out there that says, look, no kidding, there's really a problem, and here's where it's coming from. You've got this subtle bias that's going to add up. I mean, there obviously there's more overt stuff going on. There are jerks out there, but I think it's exactly as Kathy just said. It's these little things that are going to add up and have a cumulative effect to the point where you end up with your tenured faculty skewed heavily male, and that's a problem.
So I'm looking forward to this. It's on TWiM, and you said it's one. It's going to be TWiV. TWiM
48. It'll be out next week. Yeah. Yeah, Jo is great. You should listen. She's really articulate. And, she has a lab. She studies bacteria, but part of it is interest in education. This is what she does. Mm-hmm. And she's been at the White House and talked to Obamas and all that because she's got a voice here. So it was pretty good. You should check it out.
Yeah, I think it's great that this is finally crystallizing.
The next story was picked by both Rich and Alan.
Yes.
Guys like this one, right?
Polydnaviruses have always been a favorite of mine.
That's how I met you, Rich. That's how I met you. We were talking about it on the study section years ago. You told such a good story.
This is, to me, one of the most fascinating stories in virology, and it showed up in TWiV 179. Actually, there was a couple of papers that inspired this story. The one that inspired it to me was a JVI paper from... Oh, now I'm blanking on it.
March 2012. Burke and Strand.
Mike Strand.
Yep.
Mike Strand's his lab. Burke and Strand. That was basically a refinement of a 2009 paper from another group that elaborates on the whole biology of the polydnaviruses. And so basically the story is that eons ago, a double-stranded DNA virus, let me see if I can do this backwards like this, a double-stranded DNA virus which has some tendency to apparently integrate into the genome, became permanently integrated into the genome of what is now a parasitic wasp. And this thing provides functions for ultimately amplifying cellular genes that are immunomodulatory genes and packaging them into virus capsid proteins to be injected along with eggs into the parasitized organism, where these things deliver the immunomodulatory genes to the parasitized organisms, and those genes then integrate into the cells of the host and suppress the immune response so that the eggs that are laid in the parasitized host can develop. I mean, just wacky.
Yeah.
Just great stuff.
I loved how bizarre this was, and yet how perfectly rational.
Now, Alan, did you come up with this other paper?
I just put in this link to a subsequent paper from, this is the team that did the 2009 science paper.
Right, okay.
And they did a follow-up, sort of a follow-up, just recently on the evolutionary timeline of insect double-stranded DNA viruses. Mm-hmm. And they date these things back to the Paleozoic era. So these viruses were evolving around the time flies and beetles were separating.
Okay, yes. And there's currently two fundamental clades, brachoviruses and nudibviruses. And what I understood from Mike, and I actually had Mike come to Florida and give us a talk subsequent to this. I can't get enough of it. What I understood from Mike Strand, which as I understand it is the case, is that, as far as we know, is that this evolution of polydnaviruses happened twice independently, at least, because there are polydnaviruses that launch off of a brachovirus integrant and those that launch off of a nudibvirus integrant. And so apparently it happened twice. I assume that according to his timeline that the polydnaviruses are younger than when these two different insect DNA viruses split off from each other. Right. At any rate, fascinating story.
Yeah, and I just think it's so virus to for the wasp to have co-opted this mechanism as a gene transfer mechanism for its own parasitic life cycle.
Mm-hmm.
I'm sure that there are many other examples of this that we don't know about because we haven't found them. And this is a good example of, I think this is the kind of study that most of the non-science public would really not understand.
Mm-hmm.
You know, why are you studying this virus of a wasp? And we really need to make it clear how this illustrates the relationship between viruses and their hosts, and
Yeah.
that we have to study this stuff. It's not crazy. It's not a golden fleece.
No. This is the extreme end of viruses adapting to their hosts.
Yeah.
It's gotten to the point where the virus becomes an adaptation of the host.
Yeah, we need to know this because that helps us to understand how they work.
Yeah.
The next story is a pick of riches.
I'm still hung up on polymorphisms here.
You can be.
Yes.
It's fine.
You know, this to me, for me personally, was one of the high points of my year. I came back from the ASV meeting, and I told this story about a dozen times. Everybody I could corner in the corridor, I had to pin them up against the wall and tell them the story. And this is Carolyn Coyne's story of the placental barrier to virus infection. And I think one of the reasons I like this, among other things, is, well, she gave a terrific talk, but also on the when she was on the panel on TWiV that we did at ASV, she told the inspiration for the story, which was she and her a coworker in her lab, both sitting culturing viruses while they were pregnant and having a conversation, looking at each other saying, you know, is it okay that we're doing this? And this spun off this conversation and they actually followed up on it. And Carolyn went and found at her institution an expert in placenta who was able to provide them with material that they could use to culture this highly differentiated, specialized epithelium from placenta and study its resistance to virus infection. And to make a beautiful story short, and by the way, this is still unpublished. And I actually had some communication with Carolyn because I said, look, this is unpublished, and I know we've already talked about it, but I'd like to talk about it on this year-end summary. Is that okay? And she said, yeah, sure. And it's been submitted to Science and is being reviewed, and hopefully will be published in the not-too-distant future. At any rate, it turns out that they were able to show, yes, these placental epithelial cells were reasonably resistant to virus infection in culture, and that if they challenged these cells with viruses and then took the media from the cultures, that media, if applied to another completely different cultured cell, would induce resistance to virus infection in those cells. So the infected placental cells had produced something that escaped into the media that could confer virus resistance to other cells. And it turned out, they showed that what this was exosomes that are membraned vesicles that were produced by the placental epithelium that could then target other cells in culture and presumably fuse with them and deliver their contents to those target cells and induce resistance. And it turns out, furthermore, that what the active ingredient in these exosomes is microRNAs. This is unbelievable. MicroRNAs that are from a cluster of microRNAs that are specifically induced in pregnancy. 19 of them that, when delivered to the target cells, induce, among other things, and this is probably one of the major things that they induce, autophagy, which is a cell process that basically engulfs and digests foreign matter that enters the cells. What an incredible story. I just love this.
Yeah, it's really nice. So the idea is that this is protecting the fetus from viruses that are in the blood, right?
Exactly. And it's a very general mechanism, works on a large number of viruses. I think she says with the exception of CMV. And as we know, CMV can cause congenital defects.
Right.
So that's certainly consistent.
I wonder if she looked at rubella.
She said that someone at ASV, I think she said that someone came up to her after her talk, and she's really happy because she'd wanted to get her hands on that and try it.
On some rubella virus, yeah. Because also crosses the placenta. So I would guess it's not going to be inhibited.
Right.
Yeah, that's a beautiful story. And when it comes out, we'll have to do it.
Yeah, I like the story, not just for the story itself, but how it developed.
Yeah.
I really admire somebody who, like that, gets an idea, and pursues it.
Yeah.
Yeah.
You have to do that. That's how science works.
Curiosity-driven research.
Yep. The next one, our 9th story, was picked by Rich and Alan.
Yes.
You guys like this one?
TWiV 200. That was a blast.
Yes. It was great fun. So we went on TWiV to celebrate TWiV 200. We all met in Boston and toured the Needle. What is that? The Northeast Infectious Disease Laboratory?
National Emerging Infectious Disease Laboratories.
There it is. Okay, I don't even know the acronym. And which is a high containment laboratory, a BSL-4 facility. And we got a chance to not only tour the facility, but using their training facility, which is a part of this, dress up in the spacesuits that they wear in these high containment laboratories and see what it was like to work in those, and interview a couple of scientists who have a lot of experience in BSL-4 and really review issues dealing with how safe are these facilities and what can they do that we actually need done. Is the investment really justified? And I came away really quite influenced by this whole thing, very positively influenced. I came away feeling, yes, indeed, they are safe, and that there's stuff in these facilities that we need to do that can only be done in such facilities. It was a very interesting experience.
Yeah, I had a very similar experience. I went in kind of skeptical. We have a bunch of BSL-4s already. Why do we need another one? Mm-hmm. And I still have that question, but it is it was very educational about the not only the importance of the work that's done there, but the care with which they approach every aspect of it and the difficulty of it, especially in a lot of it must be significantly harder in the older BSL-4 facilities that are not as large or as well developed. So I found
one of the most interesting parts of the conversation was we were talking to Elke Mühlberger, who is one of the scientists who works in these facilities, who actually has had a 20-year history of working in high containment facilities. She was trained in Marburg, right?
Mm-hmm. Yep.
Where one of the Ebola viruses was initially discovered. And one of the most revealing things that she said was that she worked, she has, I think, 2 kids, and she worked through her pregnancies in a laboratory, and she would work in the BSL-4, the highest containment level, while she was pregnant, and did so. That she was working there the day before she delivered her child, but not in BSL-3, the level lower, because she knew that in BSL-4 she would have no contact with the agent, whereas in BSL-3 or 2, the containment efforts are to make sure the virus doesn't get out into the environment, but it doesn't necessarily ensure that the investigators themselves are completely protected from the agent. So I found that fascinating. She finds BSL-4 safer than BSL-3 or BSL-2 in some respects. Safer for her anyway.
Well, this whether this facility opens or not is probably going to be determined in 2013. The NIH is conducting a risk assessment, and I'll put a link in the show notes. You could look at those documents.
And if they don't open the BSL-4 part of it, they could rent it out as a movie set.
That's right. A very expensive one.
Yes.
Speaking of movies.
Yes.
So we made a movie there as well, besides the TWiV 200, and we've been editing it. The first draft came out at 1 hour and 45 minutes, which is, we really like to get it just under an hour so that people can show it in classes and other settings. So we're working on that. And in addition, we had to send the draft to the security people at the Needle, and they requested that some parts be redacted. I'll use that word for security reasons. So we're working on that. We hope to get it out in a couple of weeks, hopefully in January. We're just working on the last cuts now.
Good.
So that'll be fun. I've watched it a bunch, and it really is fun. So you'll enjoy that. Story number 10 is from Alan, who really liked epidemiology.
Yes, I did. And specifically the two shows that we did where we had epidemiologists on, TWiV 169 and 178. 169, we had Michael Walsh come in and essentially give us the crash course in epidemiology. And then 178 was T. Sharp. So Tyler Sharp from the Epidemic Intelligence Service came in and talked about what the CDC's frontline troops, so to speak, do actually in the field. And I found these just enormously educational. I mean, I've had kind of a crude working understanding of what epidemiology involves. But to actually have people who are in it, one who's working at the data level and one who's working on the ground level, and explaining how this field is actually put together and what it involves was just fascinating to me.
Yeah, we need to really do this more often. We should have Michael and others come more frequently because it's an important component of infection, obviously.
Yeah.
Understanding the epi. We don't do it enough. We work from our strengths, and that's not one of them, but we have to get broader.
And the statistics of it, too, I think we should brush up on that a little more.
There were a couple of especially fun parts of Tyler Sharp's show in particular, and I think they're linked in that show. So was that 178? Yes.
Yeah.
How Tequila Mosquito. Give me a break.
You don't like that title?
I think it's wonderful. Are you kidding me? He had a blog of his experience in the Marshall Islands.
That's right. Yeah.
Yes, here it is. It's linked in that episode, Marshall Islands Dengue Outbreak 1 and 2. And that blog is a really, really wonderful description of what his experience was going fairly inexperienced, going to the Marshall Islands and trying to control this epidemic.
Yeah.
Really nice writing.
Yeah, it was cool. All right. Story number 11 is also from Alan.
Yes. So what I did, I was looking at all these episodes, and Rich, I think, had the same problem we probably all did. Gee, what do we pick as 12 stories? And so I hit on the idea of cheating. I decided
it's the Kobayashi Maru test.
Yes. So I said, I'm going to declare a theme, and I'm going to pick a whole slew of episodes that are around that theme. So just running down these, we've got TWiVs 173, 195, 196, 198, 199, and 204. There was a real run of them there. And the unifying theme here is virus hunting. These are all examples of people going out and looking for novel viruses. Either they started out looking for a novel virus because there was some strange thing going on with wildlife or people, or they maybe were looking just generally looking for anything, and they found novel viruses. So TWiV 173, we talked about a new influenza virus in Guatemalan bats. And that was kind of cool because we don't think of bats really in the flu ecosystem, and yet here you've got this influenza virus in them that just turns up when you survey them. 195, this is a little bit of a stretch, but I found this episode fascinating, was Ken Stedman, who samples these boiling springs and came up with this complete freak of a virus, this duck-billed platypus of a virus. It's a DNA virus that has the capsid of an RNA virus. And so I like the freakishness of it, and it comes out of a boiling spring, but also that, here's a guy who takes his virology into the field. and goes out and looks just in the environment, what's living in there. And of course, you go looking for viruses, because if there's life, there are going to be viruses. And he found a really cool one. So I thought that was neat. And then the Joe DeRisi episode 196, that was just somebody came to him. They were concerned about the health of their pet snake, which, I guess he buried that at the bottom of his desk pile, and then he was looking for something one day, and he found it, and he said, gee, maybe I should look for viruses in these snakes. And he found there's this novel arenavirus in snakes that have this devastating disease called inclusion body disease. So there's the whole story of just the pet owner concerned about her pet snake, and then the next thing you know, you're discovering this virus that's potentially involved in a disease in snakes. And again, it comes to this sort of curiosity-driven research. You go and look for it because it looks like an interesting problem. And right now, I don't see any clinical relevance of this, but it's hugely relevant to the pet industry, and could be an interesting pathogenic process. And I think what's driving all these virus hunting studies now is the availability of the tools. Mm-hmm. So you've got, you've got the genomic sequencing, you've got all the molecular biology that's fully matured at this point, and you could do quantitative PCR, and you can do all sorts of analyses that you couldn't have done even 5-10 years ago. And so now you can actually go after this stuff. All
right. So, Alan.
Yeah.
Of all those, which is your favorite?
Which is my favorite? Oh, geez.
Just pick one.
Yeah, I should have known you were going to do that. I would say either the snake story or TWiV 198, the harbor seals.
Okay. How about you, Rich?
I'm going for the snake. Once again, because of how it happened.
Right.
Somebody's got a sick pet snake, and the DeRisi lab, Joe in particular, goes,
Yeah.
What about you, Kathy?
Well, I had picked 204 as one of mine, but I had more than my share of picks. This is the one from Skip Virgin's lab with a whole team of investigators. So this answers a question we had from a listener. This is definitely team science, where they've got people who know how to do the genome sequencing and the virology and the bioinformatics and all of that. So this is the story where they found pathogenic SIV infection in rhesus macaques is associated with an increased virome. And this was particularly interesting to me because we have a whole cluster of people in my department who are concerned with the microbiome, and to them that's defined, that is the bacteriome. And while the bacterial populations in these SIV-infected non-human primates didn't seem to change, what they did find in this Skip Virgin's work was that because they sequenced the RNA as well, they could look at the RNA viruses, and they found a whole slew of viruses that are particularly associated with the enteric disease that's a hallmark of these immunodeficiencies, SIV and HIV. So 32 previously undescribed enteric viruses, including mucosal adenoviruses and some parvoviruses. And so to me, that was just really cool, and allowed me to say na to the bacteriome people in the department.
And also, that was a virus hunting twofer.
Right.
That was the isolation of the novel coronavirus. Yeah. It's a little more classical virology, but also a new.
Yeah, that enteric virome is beautiful, and I'm waiting for the human study now, which I'm sure we'll see. The corona, by the way, they've identified a few more cases there in the Middle East, but it hasn't really taken off as people had feared it might. Right. You guys added one more?
Yeah. There's another one, TWiV 183, where they did the virome of bats and came up with a slug of viruses, and in
Right.
particular, a whole bunch of new paramyxoviruses.
Very cool. Very cool. Yeah.
Right.
All right, and our final story, number 12, from, I don't know who picked that, Rich?
Well, I think both Alan and I picked it both Alan and I liked it various times. This is the Pox accordion story, where this comes out from Harmit Malik's group, which, by the way, your interview with Harmit Malik, I thought was also on my pick list, and maybe I'll have a chance to talk about that a little later. I thought that was just a wonderful episode and a wonderful interview. At any rate, what they did here was to put selective pressure on pox viruses. They made a host range pox virus by knocking out one of a pair of host range genes that had the effect of putting a lot of selective pressure for modification of one of the gene they didn't knock out, and the virus's response was to amplify that gene into a number of copies so that it was able to regain the ability to grow on the restrictive host by basically a gene dosage effect. And then, interestingly, among those amplified genes, one reproducibly would mutate to a different form that all by itself could overcome the restriction. And since it could do it all by itself, you no longer needed the gene dosage effect, and the amplification would then collapse down to a single gene with this mutation in it. So it was a wonderful model for microevolution in response to a challenge in the environment that involved a gene amplification and then a mutation and then a collapse of the amplified genes, leaving the mutant gene intact. It was a really nice story.
And I just love the evolutionary elegance of this. You know, you've got a selective pressure that shows up, and so the virus, rather than just mutating what it has, it inflates a section of its genome and expands it, and then starts mutating stuff within that. So you essentially, it's like working with, I don't know, a Photoshop file or something, where you open a copy of it and you work on that, and if you screw it up, you still have your backups. So it's inflated this thing, it's duplicated that section multiple times, and that provides the effect that it needs. But then subsequently a mutation allows it to do that with a single copy, so then it deflates the thing back down to the single version, and it's just really, really cool.
So the other nice thing about this is that they coined this term genomic accordions, which really, I think, gave us a nice visual. And this was the first time that I had seen a paper flick, which is a video abstract of the paper. That was in Cell, and it's accompanied by the song "My Friend Nels is a Cellular Biologist," which is a tune that stuck in my head for a while today again after I watched it. So
we should point out that the first author on that paper is Nels Elde, who was a postdoc in Harmit's lab, and then and now has his own lab where he's continuing this stuff.
Right. Yep. Very cool. Well, there you go. Those are 12 stories, and they're all great.
Let me go back and make a correction. I was talking about, I get the terminology mixed up. Back in the polydnavirus story, I was talking about brachoviruses and nudibviruses. Actually, I believe I should have said brachoviruses and ichnoviruses.
Okay.
And I don't know where the nudibvirus term comes in. I think they are both types of nudibviruses. The phylogeny I'm not completely sure of. At any rate, they'll figure it out.
Well, the letters are already pouring in, Rich.
Oh, yeah, right?
Sorry.
I'm gonna get email.
Too late.
It's going to be a bunch of corrections. All right.
All right, let's do a couple of emails here. The first one's from Chadwick. Why don't you read that one, Kathy?
Chadwick writes, Doctors TWiV, let me start by saying I love science. I love science so much I download podcasts like these and listen to them religiously. I also love science so much that I am 4 years into my PhD in molecular biology. How's that for vague? I guess you could call my specialty endothelial inflammation. I love my PhD program. I love my research. I love my lab. I love my boss, as grumpy as he is. I love my fellow graduate students. I love scientists. I love it all. I even loved prelims. I hate failed experiments, but the awesomeness that is science makes up for failure. However, the thing that the awesomeness of science does not make up for is being away from my wife and 2 kids. My 3-year-old son recently said, "Daddy's home is his work." I, pardon my French, am working my ass off for my PhD. I don't like to call it work because I love it, but I have yet to find a better term. Additionally, everyone around me is also working their ass off, no matter their career stage, and they're better than me. So in order to be at least as productive as them, I have to work even more. I've been doing science for quite a while now and have yet to see successful scientists not put in 60-plus hours a week. However, I have seen unsuccessful scientists put in 40. I chose science because I enjoy putting long hours into it, but after I got married and had kids, I am now struggling with the balance of being a good graduate student and being a good husband and father. My dad always said, "These are the days you will work the hardest in your life." But as I look at my boss, a successful scientist that puts in 12-hour days at least 6 days a week, I do not see the road getting any easier. My question for the panel is this: can you be a successful scientist and work a reasonable amount of time? I understand that some weeks may be harder than others when grants are due and papers are in review and such. But I mean in general, can someone survive in science as a PI by only working 40 to 50 hours a week? If you do think it is possible, how? Do you have examples? I have an idea, but I am curious if you think the people that review grants would fund a researcher this way. My strategy would boil down to 2 main points. 1, be the best at something. 2, collaborate a lot. I figure if I'm the best at something, then everyone would want to collaborate with me and we could trade experiments. Then we would be able to get more papers out with less failure than normally comes with troubleshooting an assay that is done infrequently. Is this a naive plan? Is it that currently funded R01 investigators already do this? Are they already the best and they're still working 60-plus hours just to tread water? Would collaborations of the magnitude necessary for maintaining steady paper flow just be too daunting to accomplish? Sometimes, to borrow a theme from Alice in Wonderland, I feel like it takes all the running I can do just to maintain good status as a PhD student. And if I want to go on to a new role in science, I will have to run twice as much as that. I don't have enough time to run twice as much. As much as I love the race, I am not willing to take more time from my family. If you don't think it is possible, do you have any other career options that may suit a PhD with as much fondness for science as me? If you do think it is possible, do you know of any good PIs that may have room for a postdoc in the next year or so that wants to study virus endothelial interactions? Thanks for your time. Chadwick. Well, can you be a successful scientist and work less than 60 hours a week, 7 days a week?
Sure.
I think the answer is yes.
Absolutely.
Yes.
No doubt about it.
I would offer myself as an example. I think I have a lifetime average of about probably 45 hours a week. And I always, partly because I didn't want to work any harder than that and partly because of a commitment to my family, I was always home for dinner. We had, I've always lived close enough to the lab so that the commute wasn't a problem. And, I used my environment growing up as my role model. I left the lab between 5 and 5:30 every day. I got in early and I got a good clean start, but got home in time for dinner with the family. And then I was in charge of baths for the kids afterwards and got them to bed and all that kind of stuff. I had my evenings with the family.
Well, I've seen other people do it too. I was a student years ago in the Blazy Lab. There was a postdoc. He used to come in at 8 and leave at 5 because he had kids at home. He was the most productive person in the lab. He's now a multimillionaire. He went to a company and did great stuff. So, and there are many, many other examples too. So I think it's just a matter of doing good work, and being focused on it.
Mm-hmm.
Yep.
Hours don't matter.
Yeah.
Actually, when I was in graduate school, some of the most successful students and postdocs were people who had kids. And it was because they would set their hours and they would plan their days, and there was a very fixed schedule. They were incredibly organized about it, and they would get a lot of work done. So it could be a motivating factor.
I'm reminded of a paper that we read for the advanced committee that I was on that demonstrated that women scientists publish as much or more as male scientists, and women scientists who have families, that is. So you might think, when you're looking at two job candidates, one has a family and one doesn't, and which one should you hire? Well, don't have the bias that you shouldn't hire somebody with a family because they're going to publish less, because that's wrong.
There is this culture that says you have to work long, crazy hours, and it's very, I don't know, it kind of sucks you in. I personally, I had to resist that because I didn't want to do that, and so I felt like I was swimming upstream the whole time. But it works out, and as if we've said on this show many times before, it's just a matter of organization and being efficient with your time.
Well, I always wanted to work a lot. I didn't feel I was sucked into this because I really enjoyed it. And I mean, our solution was to have kids late in life, which a lot of people do, and that helps a lot. But then when we did have kids, my wife is also a scientist, and she worked, and she went home to have dinner with the kids, and and I stayed at work. So I have some regrets there, but that's another solution, which you can't obviously do. But I wanted to work the hours, not because I felt I'd be more productive, but I really liked it. Mm-hmm. But I don't think the difference between 40 and 60 hours is going to make you a great scientist. I think just, you love it, do your experiments, pick them well. And one thing that bothers me is that you're comparing yourself to other people who you say are better. And I just don't think you can tell that. You may think they're better, but if you love what you're doing, you're fine. Don't worry about it.
So this is where I wanted to talk about your interview with Harmit Malik, because there was maybe it wasn't so subtle, but what really struck me in that is that as he described his career, at each stage in his career, his motivation was his curiosity about whatever subject was at hand. And he went places and did things not with his success in mind, apparently, but because he wanted to solve a particular scientific problem.
Yes.
It was the purest kind of motivation. It was just wonderful. And that's where your focus ought to be.
I agree.
And I think
agree.
I think that's good general career advice.
Yeah.
Because if you try to pick, if you try to say, oh, well, I'd be more successful if I worked on X or Y, or, this is the field that everybody's rushing into, so I should rush into it too. You may succeed, but you probably won't find it as interesting as you would if you just looked around and said, what do I want to do?
That reminds me of the Steve Jobs biography, where he was talking to some marketing specialist in his company at one point who said something on the order of, what do the people want? And Jobs said, what do you mean, what do the people want? We don't give them what they want. We tell them what they want.
Yeah. Right.
I think what Chadwick has is the real passion. I mean, he started out, how many times did he say love in this first paragraph? That he really loves science. And you can love science and love your family and make it work on, both ways.
Yeah, follow the science that you love and make time for your family that you love too, and they will work together. And if you end up being less successful in your career, at least you're doing what you love.
Right.
The idea of being the best at something, I wouldn't worry about that. I think you should, as we said, just do what you're interested in, and everything else will come. You don't have to collaborate. There are many, many successful people who don't collaborate. I think it's great to collaborate, and it's probably what we're going to need more of going forward. But I wouldn't say I have to collaborate in order to get my career going. No.
And this is not pole vaulting. You don't quantify success by saying, oh, I had this many papers and somebody else had fewer. You do the science that interests you, and that's what will make the job rewarding.
Yep. And one more thing, I would try cutting back Work 50 hours for a couple of weeks. See how it works out. See if you can focus and get your work done and go home.
And I think Rich's suggestion about scheduling it for being home in time for dinner. That's a good strategy. And if that means you need to show up earlier, then show up a little earlier. And if that means you work 50 hours a week instead of 60, then work 50 hours a week. See what happens.
Yeah, and you plan it so that when you arrive in the laboratory, you know exactly what you're going to do. And you know exactly where the rate limiting steps are.
Yep.
So you know what to me, it was always a challenge, kind of a game, to see how much stuff I could get into a day. And one of the most important things is getting the long incubation started before you even make the coffee or turn on your computer, you know.
Yeah, don't get sucked into reading email first thing.
Yeah, forget it.
All right.
Good luck, Chad.
Good luck, Chad.
Yes,
Yep.
good luck.
Let us know how it works out. Next one's from Len, who sent us a post from ProMed Mail, which is via SIDRAP, which is about the evolution of influenza H3N2 viruses since 1968. It's based on a paper in PNAS. Interesting story where they have looked at the evolution of receptor binding properties, and these viruses have, they're originally avian hemagglutinins, and they evolved to interact with human sialic acids. But since then, they have lost their affinity for human sialic acids, the alpha-2,6 linked sialic acids, which are preferred by human strains. Loss of avidity for the human receptors. And this is really interesting because they say at the end this may be linked to the less severity of infection. So apparently the H3N2 viruses have caused less and less severe influenza over the years. But of course, this year, at least in the U.S., the H3N2 viruses are predominating among all flu isolates. So they're very successful despite this reduced receptor affinity.
Right. That was my question, because we had just talked about that last week on the CDC webpage, that there's this abundance of H3N2 viruses.
Right.
I conclude that whatever they're measuring doesn't matter as far as circulation in humans goes. I don't know about disease. I'm not sure about severe disease this year. But, they're using these glycan arrays on plastic to measure affinity. Maybe that's not all that matters, right?
Mm-hmm. Right.
Receptors, what do they matter?
Who cares?
The next one is from Shane Allen.
Shane writes, "Hi, Vincent and crew. I'm a layperson with a keen interest in science. I've only recently discovered TWiP, TWiV, and TWIM, and enjoy them all thoroughly, although TWiP is the easiest for me to follow. I really enjoy the back-and-forth banter between you and Dick. I'm a little confused about viruses, and I have a couple of hopefully not stupid questions. First, if a virus infects cells in the body and integrates with the genome of the host, do they then become endogenous, or is it only if it infects the cells like sperm and egg cells? Second, if a virus integrates its genome into a random location of the host's genome, won't it break the section of DNA that it gets inserted in the middle of? I would love it if you could do an episode for new listeners with an introduction to virology or even cell biology. If this has already been done, you can recommend a suitable source for this information. It would be greatly appreciated. I've started watching Monsters Inside Me, the TV series Dixon mentioned in one of the TWiP episodes with my 11-year-old daughters, and due to my newfound knowledge on parasites and viruses, I'm sometimes able to name the parasite before they reveal what it is. Keep up the great work. Cheers from Australia." And so these are not at all stupid questions. These are, in fact, kind of important questions. So if the virus integrates in the genome of the host, but it doesn't integrate in the sperm and egg cells, the germline, then it's not going to be passed along.
Right. Endogenous refers to germline integration.
Right.
Yeah.
And on the second question, if it integrates randomly, it will temporarily break that section of the DNA, but then that'll be repaired. So it doesn't break the DNA. However, it can break into and disrupt a gene.
Right.
Which is one issue with integrating viruses, is that they can cause genetic disruptions that lead to subsequent pathogenesis.
Basically, it has to break the genome in the process of integrating.
Right.
Okay. and it winds up inserted in the site where the breakage happened, and as has been said, that could cause a problem.
Right.
I think this question number one, keen interest in science, layperson with a keen interest in science, just the understanding that there's a difference between what we call somatic cells, that is not germ cells like sperm and egg cells, and sperm and egg cells. That little bit of insight, intuition there is profound.
Yes.
Really good question.
Yes, excellent questions. And for basic primers for virology, there is, or at least was, a series of TWiV episodes that we refer to as Virology 101. And perhaps one of our resolutions for 2013 should be to get back to that.
There you go.
TWiV New Year's resolutions.
All right.
How about that?
Great.
All right, in January. How's that?
Right.
All right.
Yeah. Okay.
There are 9 Virology 101s, and there's a whole separate tab at the top of the TWiV page. But Vincent, you might put just the link in this to these
Yeah. I'm trying
Virology 101s.
Yeah, I'm trying to figure out how to make some of these things more obvious, and I'll work on that. But yeah, we'll put a link in the show notes. The next one is from Jason, who sends a link to a Times article about the Pakistani immunizers being killed. I don't really have anything specific to say, just to have a terrible heaviness in my heart.
Right.
Jason is a polio person from Australia. And Rich, can you take the next one from Matt?
Matt writes, Dear Alan, Dixon, Vincent, Kathy, and Rich, in random order supplied by a random number generator, and he's got a footnote to that side. MATLAB command rand perm. Jeez. Talk about nerdy. It is a drizzly 41 degrees Fahrenheit, 5 degrees Canadian here today in Bar Harbor, Maine. I write you because I came across this article in Slate magazine today that addresses flu vaccine skepticism. Preliminary data suggests that this will definitely be of interest to your listeners, n equals 1. I suspect that many of you are aware of the mainstreaming flu vaccine skepticism. The author of the article is Dr. Darshak Sanghavi, a pediatrician and medical expositor. He starts the article by cataloging several pieces in mainstream news and opinion sources promulgating skepticism of flu vaccine efficacy. He then attempts to dispel the skepticism by taking a historical perspective on infection control measures and shows how our current flu efforts rhyme with successful control efforts in the past. He goes on to discuss the idiosyncrasies of influenza and details how our current efforts to control it, vaccinations, Tamiflu, mandatory reporting of flu in children, etc., are both a work in progress and also measurably better than the alternative. Of particular interest to me is that he chose to cite primary literature to support his claims throughout the article, giving links, for example, to the British Medical Journal and the Morbidity and Mortality Weekly Report. This provides a good set of source material for framing a conversation about appropriate policy. But more broadly, this style of exposition shows tremendous respect for his reader's intellect, and in my opinion should be a model for science journalism, especially journalism surrounding public health matters.
Hear, hear.
Diseases are scary, and people, when confronted with them, will take the time and effort to get informed. Talking to people through primary sources demystifies what scientists think and why they think it. That has real power to change minds. So, at this point, I will conclude by thanking you all for the Twix series and your collective commitment to primary source news and opinions on pathogens of various types. I realize that you are not always discussing public health matters, nor do I think you should, but high-minded conversation about public health requires sophisticated understanding of all the issues. Influenza itself reminds us that controlling infection requires understanding multiple levels of organization, from the molecules causing disease to the herds of humans swarming around the planet sneezing on each other, vaccines and Tamiflu in the small, herd immunity in the large. Each pathogen is different and complicated. Einstein apocryphally implores us to make things as simple as possible, but no simpler. An adequate public response to complex pathogens requires an adequate public understanding of their complexity. As such, there is no substitute for adequate information and likewise no substitute for TWiV, TWIP, and TWIM. My listener pick of the week is an article I found in Slate Magazine today. Oops, recursive email. I hope this finds you well. Matt, what a wonderful email.
Nice.
Outstanding.
And
just really terrific.
This last sentence, as such, that should be on the front page of the website.
Yes.
It's like a product endorsement. There's no substitute for TWiV, TWIP, and TWIM. I love it. Very nice.
And I agree with him. I took a look at the Slate article, and I clicked on a couple of the links in it, and it took me to the primary BMJ source and so forth, and I really admire that.
I really, really wish all the mainstream publications in news media would do this. Because so often I'll see a story and maybe I didn't see the press release or I didn't pick up on it or whatever, and I'll see some news story about a scientific paper, and they'll say, you know, the new paper that came out such and such time is by so-and-so's lab, and I'm thinking, where is it?
Yeah, never.
Give me the darn link already, and usually they don't.
Yeah.
And you know, a lot of these online journals now are written with author summaries and et cetera that are actually quite readable, and they're much more accessible to the public. So there's no reason not to include the primary literature.
Well, and even if the primary paper is completely impenetrable to non-specialists, let your readers decide that.
Yeah.
Right.
So you think the New York Times should do that, Alan?
Absolutely. All their online stories should link to the primary research they're talking about.
So my wife was reading a front-page Times article about some Merck drug the other day, right? Mm-hmm. And she's reading it. She reads the whole thing. She goes, God damn it, they don't even say how the damn thing works. What piece of crap is this article? Right. And here she was she's not her drug. She wants to know what's going on. Right. And they don't even, if they had a link, that would help, right?
Right. I find myself doing the same thing. I read all of these popular press articles, and I'm doing my own research to find out, you know, just like whatever drug it was we talked about earlier, the poly-IC. I had to look that up and figure it all out.
Yeah, yeah, I'll tell you. All right, let's wrap it up with some picks of the week, starting with Kathy.
Okay, well, I guess this is a pick for the next 110 years, not just pick of the week.
So you're done for a while.
Yeah, it's Popular Mechanics, and they're celebrating their 110th anniversary, and they have made 110 predictions for the next 110 years.
That's pretty bold.
Yeah, and they have the first ones are just what's going to happen in the next 10 years, and so maybe those are the most accurate predictions. And it's just fun to look at. And then they have sections on 10 things that will remain the same, and then later they go into some predictions further out from 2023 to 2062, and 10 things that will disappear, and it's house keys and your wallet and gaming consoles and so forth. And then they kind of have an executive summary at the end of the Popular Mechanics Brain Trust says, within 20 years, self-driving cars will hit the mainstream market. Within 30 years, all-purpose robots will help us with household chores. Within 50 years, we'll have a colony on Mars. Within 60 years, digital data, texts, songs, etc. will be zapped directly into our brains. Now, you know, that could save hours per week if the science literature would just go right into our brains. And then their prediction for within 100 years, the last gasoline-powered car will come off the assembly line. So
I found that some of this I found extraordinarily optimistic, which I really liked.
Mm-hmm.
You know, that there will actually be solutions to problems that seem impenetrable now.
Mm-hmm.
Hey, one is up our alley, that vaccines will wipe out drug addiction. We did a story on that, remember?
Yeah. And did you see this one for Dixon?
Which one's
Vertical farms will feed cities.
Oh, look at that. Cool.
What I didn't see, I didn't look at this real closely, but I was thinking earlier as we were talking about some of the genome stuff, that personalized medicine I would say it's going to be, everybody's going to have their genome sequenced. The bioinformaticians are going to figure a lot of this stuff out, and there will be personalized medicine. They're going to tell you, what things you're more susceptible to, et cetera.
Hey, how's your genome coming there, Rich?
Slowly. I think they've taken samples. I spit in a tube and sent it to them.
Yeah.
But I haven't seen any sequence yet.
Okay. Rich, what do you have for us?
Oh, geez, I'm up. I'm going backwards in time. I wonder if any of you guys remember this. A movie produced by Bell Labs in 1956 called Our Mr. Sun. This had a profound influence on me when I was growing up. Apparently, in doing research on this, Bell Labs, which used to be AT&T and is now some French communications company and is quite an establishment as a research lab, having produced something like 7 Nobel Prizes over the years.
Yeah.
In the 50s and 60s, produced a series of 9 films that were intended to communicate science to the general public. And the first of these was a film called Our Mr. Sun, which is a combination. It was produced and directed by Frank Capra, or actually written and directed by Frank Capra, and had a couple of other stars in it. Lionel Barrymore plays Father Time. And it uses science and animation to try and communicate science and scientific thinking to a general audience. And apparently, I watched these films in school. Bell Labs made the 16-millimeter films available free to schools. So this now you can watch on YouTube. I'd been wondering for years. I had vague recollections of it, and the other day I watched the whole thing on YouTube, and it's quite entertaining. I have some issues with some of the stuff towards the end, but nevertheless, it's a classic. Put it that way.
Cool. Did it bring back memories, Rich?
Oh yeah, sure. I was 8 years old when this was made. I probably didn't see it until I was 10, maybe, something like that. But I remember this one, and I remember the next one, Hemo the Magnificent. I don't recall any of the others in the series.
Alan, what do you have?
I have something that was recently announced by the NCBI, the home of PubMed. This is called Pub Reader, and I suspect a lot of our listeners will get a lot of use out of it. I suspect I will get a lot of use out of it. Essentially, what it is they've added a set of features on PubMed that allow you to read on a variety of platforms. So instead of downloading the PDF and just reading through the PDF on your computer screen, and then maybe you go and you want to read on a tablet or a smartphone or some other device, but the PDF is essentially unusable on that device. Now, instead of doing that, they've got this set up so that it properly reformats the page for the device that you're using to read the paper.
This is so beautiful.
And you can, I gather, I think if you sign in, it will mark where you were.
Yeah.
So I'm not sure I'll use that, but the idea of being able to put something on my phone, and I tried it. That's the extreme case, is can you read a research paper on your phone? And so I tried it, and it works. I can look at a paper on my iPhone, and the text is a good size. I can resize it. I can flip the pages like I would flip the pages on an ebook on the phone. And obviously it'll work on an iPad or some other tablet, and it'll rearrange the text. So it really finally makes the research literature accessible through the screen that you happen to have handy.
This is beautiful. One thing I've always had trouble with reading on a screen is if the images are way at the end of the paper, like reviewing a paper.
Yes.
You have to scroll back and forth. With this, they put the thumbnails at the bottom of each page, and you can scroll over, and it pops up the image. It's really nice.
Yeah.
I really like that.
Yeah, so anytime you're looking up a PubMed citation, up at the top, one of the options now will be the Pub Reader format. And obviously, if it's in a if it's behind a paywall and you don't have access to it, you've still got that problem. But for open access or stuff that you have access to, you should be able to download it this way.
Our tax dollars at work.
Exactly, yes.
Beautiful. That's really nice. My pick is a article from Nature. It's called 366 Days, Nature's 10 People Who Mattered This Year. And we know two of them. One is Joe Handelsman, who they call the bias detective. And that's, of course, for that work that we were talking about earlier. The other person on the list is Ron Fouchier, who they call flu fighter. Although I would not call him a flu fighter. He doesn't really fight flu. He works on
Studies it.
Studies it. And then the other people are various scientists. So I thought that was pretty cool. But mainly because Joe's there and I know her. So anyway, that's Nature's 10. We have a listener's pick. Matt gave us that article from Slate.com, which we'll put up as our listener pick of the week for this week. And that brings an end to TWiV 213 and an end to TWiV for 2013.
2012.
Is it 2012? Yeah, I'm looking ahead, huh?
Yeah.
You can find TWiV at iTunes at twiv.tv, where you can find our show notes. Do check them out. They are pretty neat. You can find us on Facebook, facebook.com/thisweekinvirology. If you do like us, the best thing you can do for the moment is to go to iTunes and rate us. Give us a bunch of stars or leave a few comments. That helps us to stay on the front page of the iTunes directory, which we quite successfully do, which is really nice. We're always up front there. We love to get your questions and comments. Send them to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thank you. This was a lot of fun. I learned some, and it was fun to review what I learned through the year.
Yes.
So I'll see you next year.
Yeah, and thank you for joining the TWiV team this year.
My pleasure.
Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Always a good time. Thank you. It's great fun.
Now you can go back to your family.
Yep. Nap time's about over.
All right. Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Happy New Year to everyone. We hope it's been a good 2012 and hope you have a great 2013. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.